As we continue with the third part of chapter 11, I want to remind you about chromosome structure, especially chromosomes that are replicated homologous chromosomes. So if we look at this particular diploid cell, we see that the red chromosomes represent maternal chromosomes and the blue one represent the paternal chromosomes. This is a diploid cell. Now, after DNA replication occurs, these replicated homologous chromosomes consist of a pair of sister chromatids that are held together by the centromere region. And as I mentioned when we looked at the karyotypes, the centromere region is not always in the very center of the pair of sister chromatids. Its position gives a unique shape to each of the chromosomes. So what does all this have to do with sex? Well, recall that when diploid organisms reproduce to produce offspring, there has to be a reductional division. And this is where this process of meiosis comes in. Our zygote, our single cell that is the result of the fusion of a haploid egg and a haploid sperm, can then grow by mitosis to produce the more somatic cells. These germline cells, these are the cells that are going to be capable of undergoing this process known as meiosis to produce more gametes. Now, the need for cell division processes to produce these haploid cells for reproduction is pretty evident. And again, this is called meiosis. Overall, we can think of meiosis as cell division with a single replication of the genetic material, followed by two consecutive cytoplasmic divisions. So think of it as duplicating the genetic material once, but dividing the cells through cytokinesis twice. Now, there will be two discrete steps to this, meiosis 1 and meiosis 2. And the end result of the two phases of meiosis will be 4 genetically unique haploid cells. And we can see this in this simple diagram here. We have a pair of homologous chromosomes representing our diploid parent cell that replicates them, that go through replication. They divide in meiosis 1 and the homologous chromosomes separate. Once they divide again in meiosis 2, it's the sister chromatids that will separate. And so you can see then how we go from 1 cell to 4 cells, and the 4 cells are each haploid. Now, the process of meiosis is very similar to mitosis. There are going to be a lot of overlapping steps with some very similar things that's happening. Be sure you note the differences as I'm pointing them out to you. And it's important because these subtle differences are going to allow for this genetic variation in cells to be produced. We're going to see that genetic variation in sexually reproducing organisms is not just produced because of having two different parents contributing. We're going to see some true genetic variation that's a byproduct of the process of meiosis itself. Now, meiosis will begin with a cell that has completed interphase. And if the cell has completed interphase, then recall that during the synthesis portion of interphase, DNA replication has already occurred. And so if we look at this slide, this represents an overview of these steps of meiosis. So following interphase, the cell will go through meiosis 1, which includes prophase 1, metaphase 1, anaphase 1, and telophase 1. Following telophase 1, the cell doesn't go back to interphase because there's going to be no replication of the DNA, so therefore no synthesis phase. There is a brief period of rest, and the cell kind of recovers itself and begins to produce some more enzymes that will allow for meiosis 2 to occur. This phase is called interkinesis, literally the portion between the divisions. Following interkinesis, we see meiosis 2, which consists of prophase 2, metaphase 2, anaphase 2, and telophase 2.